The overall goal of the following experiment is to identify microRNA targets amongst a library of 3' untranslated regions, or UTRs. This is achieved by co-transfecting HEK293 cells with a microRNA expression plasmid and a plasmid containing a luciferase reporter fused with a 3' UTR. To facilitate large unbiased screens, transfections are performed in 96 well plates. As a second step, the cells are subjected to a dual luciferase assay, which measures post-transcriptional regulation of the 3' UTR by the microRNA. Next, data from multiple replicates and multiple plates are analyzed in order to identify high-confidence microRNA targets amongst a pool of hundreds of 3' UTRs. Results are obtained that show each microRNA targets a unique set of 3' UTRs based on translational repression as measured by the dual luciferase assay.
The 3'LIFE assay can answer key questions in microRNA biology by identifying a large number of target genes for a microRNA of interest, yielding data to direct downstream validation experiments.
"24 to 48 hours prior to transfection, seed a sufficient quantity of HEK293 T cells based on the number of 96 well plates to be transfected. Each 96 well plate requires 9 million cells. In this demonstration, 2 well-characterized microRNAs will be screened against a panel of 275 human 3'.